Hello, classmates. My name is Junyi Lai. I'm a medical oncologist at the Department of Oncology at Taipei Veterans General Hospital, and I'm also currently assistant professor at the Institute of Clinical Medicine at our university. I am an oncologist. In my daily practice, I use a lot of anti-cancer drugs, including biologics, which we will introduce in this lecture. And for the recent years, there has been a wave in biosimilars, which is a cheaper or more affordable alternative when compared to the biologics drugs that are currently still on patent. And um, the biologics is also proven to be as equally as effective as the biologics. So we will all get into that in the next 1 to 2 hours. So my apologies that I could not be there in person due to a scheduling conflict, but I have recorded this video, which will go through all the lecture, and I hope that there will be informational for you. And in the last slide, I have listed down my email. So if you have any questions or you want to discuss any points, then I'm very happy to discuss with you over email regarding any issues or any questions you might have on biologics. So we will get into the whole scenario of first the biologics and then the biosimilars, the development and the current landscape. And this is actually quite an important issue, especially in 2025, where all the new drugs, the prices are higher and higher. So actually, yesterday I was uh, called to join a meeting at our hospital to discuss the current use of the current use of biosimilars in our hospital, where our hospital hopes that more people or actually more physicians could utilize biosimilars instead of biologics. Currently, roughly about 10-20% of the biologics are being prescribed using biosimilars, and our hospital hopes that the number will go to 30%. So as you can see, this is actually a very real need and trend in terms of the current situation of medicine. And as you will mention in this lecture, many countries and many other areas, such as like Europe, they have already widely embraced the concept and utilization of biologics. A lot of uh, a high market share of biologics occurs in Europe. So we are getting to that in the next about 1.5 hours. So hopefully this lecture will be helpful for you. I'm a little quick introduction of myself. I got my MD from Yangming University, so I'm an alumni of our school. And then I went to did do internal medicine residency at the Veterans General Hospital, and then I got interested in basic research, so I went to the Scripps Research Institute PhD program in San Diego, in the USA. So I got my PhD from US, and then afterwards I came back and completed the oncologist training, and then became a attending physician at the Veterans General Hospital. So you can say that I'm an alumni from our school and from Veterans General Hospital, and now I'm back uh, as a faculty and as also as a attending physician. But it, my during my PhD, I did full time in Scripps Research Institute, which is one of the top 10 biological science program for PhD. So in my heart, I consider myself a molecular biologist, a basic scientist as well.
So if you're interested in science, and our lab focuses in breast cancer, uh, especially molecular biology, especially epigenetics. So if you're interested in breast cancer research, in cancer research in general, or if you have any questions, I will be happy to help you answer. Let's say regarding like job opportunities, or like research, career development, especially in the cancer aspect. So this is my web page. So I have the left is the physician web page at a veteran general hospital, and the right is our lab web page. Haven't updated it in a while. So we our lab recently has some great results, and we're hoping that our results can be published this year. And it uh, so far the results are pretty interesting. Okay, so back to our today's class. Our topic today is the overview and the introduction of biosimilars. So we will start with the description of biologics, and then we'll go into biosimilars for the second half. Um, so why, if the top title is biosimilars, why are we talking about biologics? That's because biosimilars is a kind of biologic, and it is extremely related to the understanding of biologics. So you cannot understand biosimilars without have a good understanding of biologics, because all the characteristics, all the profiles are related to biologics themselves. So let's start with the definition of what are biosimilars. A biosimilar is a biologic that is highly similar to and has no clinically meaningful differences from another biologic, which is FDA approved. So here we can see there are several keywords. There needs to be a biologic that is already FDA approved. Biosimilar is something that is similar to this biologic. So if the biologic that it is similar to is not FDA approved, this cannot be classified as a working biosimilar. You can say it's highly similar, but it is not by definition biosimilar per se. Biosimilars are should be given the same way, the same route of administration. So if the original biologic is given subcutaneously, the biosimilar has to be given subcutaneously. This has the same strength and some same dose, same potential side effects, uh, and in general, the treatment benefits, the treatment efficacy should be similar. And this is important. It cannot be significantly better than the biologic. It can be similar within the range of statistical non-difference. Uh, it cannot be better or worse. So this is the definition of biosimilars. So highly similar but not identical to the biologic. So now I guess the question is, why can it be highly similar when it is not uh, identical? I mean, why do we not allow it to be identical? Uh, so this will be answered in the following slides, and this is important for the understanding of biosimilars. And the reference biologic that it is based on is called the reference, or called the originator, or called the reference product. So there are several names, but basically the concept is the same. It is a previously FDA approved, well, strictly speaking, not only FDA. You can also be EMA, which is the European equivalent of FDA, or the CFDA. Anyway, from the regulation agencies, FDA approved uh, biologic. Uh, if your this drug is similar to the biologic, then it can be called the biosimilar. So for the next uh, probably one third or half of this class, we will be talking about biologics because it is, as you can see, extremely important to understand biologics before we talk about biosimilars. So biologics are created by definition by living cell, microorganism or mammalian cells, living cells, and biologics mo- are large molecules. Most of them are proteins or polypeptides. So here's a table comparing small molecule and biologics, which are two major categories of medicine. Small molecules carry a small molecular weight, 0.1 to 1 kd. Biologics are larger by definition, larger than 1000, but 1 kd, 1000 daltons. But importantly, this is not uh, the like the normal range of biologics. Usually, biologics are much larger. Let's say uh, 150,000 or maybe more. So biologics are complex and much larger than small molecule. Small molecules are stable to heat, to a lot of physical environmental stress. Biologics, basically just think of biologics as proteins. They're sensitive to heat, they're easily degraded. Um, just think when you order antibody for Western blot, it comes in packed with ice. It cannot be heated up. So you can see that biologics, just think of it as proteins. Small molecules will often have off-target effects. So because of the size, it might be promiscuous binding to other different receptors or other different proteins, while biologics are highly selective. They have a very usually, especially when you think of it as protein or antibodies, they will have a specific binding target, let's say a receptor. So if you think of it as a binding target or binding receptor, then it will be selective. It will be the binding process or the interaction will be highly selective. Biologics have higher immunogenicity because it's a protein and it will more likely be identified by the human immune system. Um, Usually, small molecules are cell permeable, again, due to the size and the biochemistry. Sometimes it may be CNS penetrant, depending on the physical properties of the size, while biologics are usually not CNS penetrant. And the distribution for small molecule is just free flowing, circulating in the bloodstream. But for biologics, uh, they will be more preferentially distributed. Again, this is just think of it as a protein. The metabolism breakdown for small molecules is cytochrome or liver renal function. The biologics will be receptor mediated. Small molecules have more drug-drug interaction, and small molecules are much more cheaper to make. Basically, the making of small molecules is uh, purely chemical. It's a chemical synthesis process. So you can have a manufacturing line set up, and this will be a machine, and you have a well-defined chemical synthesis route. Usually, um, the purity will be very high, and each small molecule will be nearly virtually identical um, to another. This is different from biologics. Every biologic is made from a cell or a living organism. So, um, just like when you are doing cell culture, can you promise that the cell culture of your cell culture today and your cell culture last week, the medium composition components are the same? Can you promise that, or do you think? that is the case? It's very likely not, right? Because even if you're very careful, you're very diligent, uh, in the you cannot guarantee that there will be no small handling errors between today and mm, a week ago. And so the medium itself might have impurities, might have some like toxic waste, and then that might change the pH a little. And then in that sense, all the cells will be exposed to a different environment, and the protein that they produce, at least the production, will be maybe altered. And you don't know if the pH will affect the, let's say, the glycosylation profile of the biologic or the protein that you're making, and that may have a difference. So very simply speaking, different batches of biologic may have different post-translational modifications. Obviously, the protein that makes should be the same. Let's say you have a CHO cell or any kind of protein producing cell line that makes monoclonal antibodies. The protein that is made, each batch is the same sequence, the same primary sequence to be exact, but the folding of the protein will be different. Theoretically it shouldn't change too much. However, the glycosylation will be different, might be different, and for every batch, the glycosylation will fall within a range. It's very likely not 100% exact, identical. So that's the weird part, and it's a part that's kind of difficult to understand. But that is why each biologic batch, you cannot guarantee that it is identical. So the most you can say, it's the same sequence, it's highly similar, and after testing, it needs to pass the test of clinical trials to show that the efficacy is similar, and that's the best you can hope for. That's what fulfills the definition criteria of biosimilar. So I've highlighted the important points, just the past minute. Um, Biologics, the activity is dependent on culture conditions, the conditions of living cells. A glycosylation pattern will determine antibody function, and the production and the manufacturing process is kind of complex, and the yield is not as high as small molecules, and it is more labile or more sensitive to environmental stresses, more immunogenic. So this is just a schematic illustrating that, as you can see here, antibodies will have glycosylation on their surfaces. That's known as the post-translational modification. And these glycosylation of antibodies will determine the antibody function. So you can have identical primary sequence, but in the end, it is the glycosylation pattern that will define the actual binding, and sometimes it will vary by a couple folds, even tenfold or more. There are some known specific glycosylation patterns of the antibody that would highly impact the binding of the antibodies. So there are several companies that are doing glyco engineering, which is known as how to site specifically or how to accurately modify the glycosylation of the antibody, and the process is known as glyco engineering. Okay, so back to the description of biologics. Um, the biologics, again, the primary sequence is the same, but the molecules may be heterogeneous. The impurity profile is batch dependent, and it is wide ranged. The regulation in agencies will need to characterize the protein target, the active agent, the API, and the surrounding adjuvant supporting agents of the whole solution. Um, Ideally, it should be as homogeneous as possible, but it cannot be strictly enforced because again, this is the production of biologics. One example, by FDA definition, blood. If you have anemia, you need blood transfusion. Blood is a biologic. Blood is not uniform, as for obvious reasons. Just think of today, there's one donor, he donates 2 units of blood. Tomorrow, another donor, he will also donate you 2 units of blood. Both of them have O blood type. Do you think these blood products will be identical? Of course not. So biologics, because of its unique characteristics, it can the product themselves homogeneous heterogeneity or differences may be allowed. Here are some categories of biologics. Again, just think of it as peptides. So interferons, interleukins, growth factors, monoclonal antibodies, polypeptides, vaccines. Um, we want to focus on monoclonal antibody because that's the most impactful biologic in current medical medicine development. So monoclonal antibodies are defined as a single species of immunoglobulin molecules by hybridoma cell. It can only recognize epitope of an antigen substance. The way that monoclonal antibodies, well, I'm sure that you more or less know the concept. So basically, you inject either mouse or rabbit with the antigen, you stimulate the immune system to produce the antibody, and then you fuse the antibody. You extract the B lymphocytes, which can produce the antibodies from the mouse or from the rabbit, and then you um, what's the word, immortalize it. So how do you immortalize it? You don't want to do genetic changes because you might alter the cell. So you uh, co-mix and create a fusion or um, a mixing of the spleen cells, the B lymphocytes, and the tumor cells. So then you have a long growing uh, hybridoma line that will continuously to make the antibody that you have just raised from the antigen. So adding PEG will cause a hybridoma and then just grow it as cell culture. This is a schematic cartoon figure. Uh, so now that you know how to make the antibody, there are many ways that you can manipulate and use antibody as an approach, a route for drug discovery in medicine. So there's a concept called phage display. So phage display, well, we don't need to go into detail today because today we're talking about biologics. But anyway, just keep in mind that monoclonal antibodies can be discovered, can be screened for their targets. And then you can raise a specific antibody for a specific target of interest, and then you can develop it as a drug. So phage display is just one of the route where you can do that. Phage display was perfected by Richard Lerner, which is who is the founder of the Scripps Research Institute, which I got my PhD from. So his work led to the discovery of Humira, which is now the most important biologic. It's not an anti-cancer drug; it's an anti-rheumatic disease drug. But it's now one of the most important biologics, one of the most best-selling, uh, highest market grossing biologic, and which is the important revenue for AbbVie. Um, so he's kind of a pioneer in the monoclonal antibody field. So biologics is cancer is a rapidly growing segment. It's now more or less half or more than half of the whole drug development market. The other half is mostly small molecules. So the market size of biologics is growing at a really rapid pace. Uh, so when we talk about biologics, especially its development in cancer, there's one company that we definitely have to mention. This is the Genentech.
uh, company. And this is the probably the most famous biotech company. If you're in bio industry, there's no way that you've never heard of Genentech. And for us in academia or in clinical medicine, Genentech is also extremely important. So it's one of the pioneer companies who started with the development of monoclonal antibodies as therapeutic, and it throughout this process and its drug discovery process, it turned out many currently approved drugs in cancer. So the drugs or the medication from Genentech are has vastly changed the outcome and the treatment landscape for cancer. Genentech is a very interesting company that it sets the precedent. It's the kind of a pioneer in the sense that it is not only just a commercial company who is all about like selling drugs and sales and marketing. It's also very heavily rooted in R&D. And in this sense, the scientists as genetics are generally noted as leaders in the biomedical fields. And these leading researchers or scientists from genetic, they participated in the success of drug discovery for all the drug portfolio of Genentech. And uh, when we talk about like universities, the prestige of the university, we often discuss or consider their scientific output and scientific contributions, right? So we know that like Harvard University, Stanford University, these are the best universities in the US. In Taiwan, you would think of National Taiwan University or National Yangming Jiaotong University, which is our school. These are very good leaders in terms of academia and in the biomedical field. So Genentech is a commercial company. However, the scientists from Genentech, because they do science at the highest level, highest caliber, so scientists from Genentech often become leaders. They sometimes they get recruited by other top universities and become a leader or a chairman of the academic position over there. So, for instance, as you can see, there are scientists from Genentech who would go to Stanford or Rockefeller or Duke to become dean or leader or president. And in this sense, these leading biotechs, these that are research oriented, are more or less a little bit similar to universities because they do a lot of R&D and they have very rigorous science standards. And what's more interesting is that there is actually a postdoc program at Genentech. So you could do a postdoc at this biotech company, and you can be assured that the scientific training and the scientific environment is of top notch. So this is a very, I think it is a kind of a role model for biotech who are doing good research in the industry. So just to very briefly mention the history of Genentech. It was founded in 1976. It was founded by a venture capitalist and associate professor at UCSF. The Professor Boyer, his important discovery is expression of human proteins by bacteria, and this led to the work related to restriction enzymes, which we are using in molecular cloning every day, and also this leads to the genetic engineering era. Um, so we're going to briefly talk about a wonder drug called Herceptin. This is a very important monoclonal antibody drug for breast cancer, and it was developed at Genentech. Originally, the idea was a very academic question. Could you discover what genes that are overexpressed in cancer? So they did a very simple screening to discover what kind of genes are overexpressed in cancer. This is a picture cartoon news cartoon that shows that the discoverers of um, Herceptin and the developers, they won the Lasker Award, which is a very prestigious award, almost close to the Nobel Prize, and they won it for their discovery of Herceptin because now at this day, Herceptin has changed so many lives in patients with breast cancer, so it is extremely important. So as we just mentioned, genetic scientists try to screen for any genes that would be overexpressed in cancer. So they discovered a gene called HER2, and HER2 is a membrane-based protein that is on the membrane of the cell, and the the cell membrane receptors that would in turn lead to intracellular signaling, and then that would give the cancer cells a survival advantage. So about 20 to 25% of the breast cancer patients would have HER2 expressing cells, and these patients would be classified as HER2 positive breast cancer. Up to today, in clinical world, we still classify patients uh, using this manner. So if a patient, the cancer cell of the patient is HER2 positive, we call her HER2 positive breast cancer. So at this point, today, in 2023, it's now very standardized treatment for HER2. But back in those days, HER2 was HER2 breast cancer was not yet very well defined. So genetic scientists came up with a hypothesis that if you could develop an antibody that would block HER2, bind to HER2, and block the intracellular signaling, it would lead to cell death of the HER2 signaling cascade. And in this sense, by adding an antibody to the cancer, you could treat and decrease the tumor size. So genetic scientists develop an antibody, a humanized antibody against HER2, and this study was published in PNAS, which is a very prestigious journal. If you can see the authorship here, you can see that all the authors come from genetic, and they even have department of protein engineering, cell genetics, cell biology. This is just similar to any university. So you can see that genetics is set up as a very rigorous or very high caliber research oriented institution. It just happens that it's a drug company, but it's still the essence is like a biomedical university or institution. So at this point, uh, the idea, the concept itself is pretty novel. They want to develop monoclonal antibodies as therapeutics. Uh, Now in 2023, this is already a well established fact. So basically, monoclonal antibodies. Well, I think probably most of the audience today I think you guys know the difference between monoclonal and polyclonal antibodies because we use it every day in our experiments like Western blot or like flow cytometry, etc. So you know that monoclonal antibody is more specific, is usually comes from a single antibody species, will have lower signal, lower noise, better signal to noise ratio. So it's specific, and when you develop it as therapeutics, it can have good results. So back to Herceptin. So as we can see, Herceptin has would then published very good clinical results, and we won't go into the details today because we're not teaching about clinical trial results here. But we can see the progression-free survival is 7 months when use Herceptin plus chemotherapy versus only 4.6 months if you only use chemotherapy alone, and the response rate and the overall survival are really good. And this was published in New England Journal of Medicine, which is the most prestigious journal in medical field. And the results that are we call it guideline changing, which means that if you have the results, it changes the guideline. Then at that point, this is basically you have changed the history and the treatment algorithm or the treatment landscape of medicine. So this is really interesting, and this is at that point that was a groundbreaking result. So the last slide, it was a successful trial, right? And that was in metastatic HER2 metastatic breast cancer. So in the cancer field, once a drug has proven its efficacy in metastatic disease, the drug company would want to try to do clinical trials in non-metastatic cancer of the same cancer cell type. For instance, like in early breast cancer or non-metastatic breast cancer. This is important because if you do the clinical trial in metastatic breast cancer, the indication from FDA will allow you to sell the drug, but only to patients with metastatic breast cancer patients with metastatic breast cancer. So if you want to use it in non-metastatic breast cancer patients, you have to do a clinical trial in early breast cancer, and that in that sense, you can now use it if you have a we call it the label, the FDA indication, then you can use it for early cancer patients. So Herceptin did that, and they still succeeded, had a very good results in terms of decreasing the recurrence rate for early breast cancer. So at this point, Herceptin dominated the HER2 breast cancer landfill. So every patient who has a HER2 breast cancer would be using Herceptin. It's either in the metastatic setting or in the early breast cancer setting. And this is again further research that emerged from Genentech. So we now know that in metastatic breast cancer, Herceptin is the most important drug, target therapy, but they pushed the concept further. They came up with a hypothesis that if they develop another HER2 drug, which they did, it's called Perjeta, and use it in combination with Herceptin, would that make a better clinical result, clinical outcome for HER2 breast cancer patients? This is a very bold and interesting hypothesis and idea for the cancer field, because up until now, there has not been no or very little cancer therapies that have two drugs that are targeting the same receptor. Because usually you would think that if you have two drugs but that target the same receptor, then when you treat the receptor of the cell would be saturated by probably one of the two drugs. But this is the first time that they proved in both preclinical models and in clinical studies that the addition of Perjeta to Herceptin would lead to further added expanded clinical efficacy of HER2 blocking. So this is very, very important breakthrough in the concept. So this is called the Cleopatra trial. They did a phase 3 study to confirm that addition of Perjeta actually made the outcome of these metastatic breast cancer patients even better. In 2019, they had an end of study analysis, and you can see, well, this is a lot of clinical study results. But anyway, just the takeaway from this trial is that the uh, the addition or the combination of dual HER2 blocking is really, really good in terms of performance and the outcome, and that leads to excellent treatment efficacy in patients, and that translates to very good survival data. So how good is the survival data? So in the previous slide, which I kind of just glanced through it, is that um, if you use Perjeta plus Herceptin, the overall survival is 56 months. So I don't know if that means anything to you. So let me break it down to layman terms. When we talk about clinical trial, we talk about survival. It's always in the terms of median survival, which means that if we say it's 56 months, that means that half of the patients would live longer than 56 months. Half of the patients would live less than 56 months. And these are all metastatic breast cancer patients, which means that from the beginning, their outcome was already really poor. So if you treat them with Herceptin plus Perjeta, half of them would live longer than 56 months. What is 56 months? It's close to 60 months, which means that it's close to 5 years. So half of the patients would have a chance of living more than 5 years. That's actually very, very amazing in metastatic or phase 4 cancer patients. So previously, if any patient has a phase 4 disease, we would tell them, oh, this is not good. The survival outcome is not good. But for HER2 positive breast cancer, because of the invention of Herceptin and Perjeta, we know that the the expected outcome, you have a 50% of chances that you can live longer than 5 years. This is excellent. And this is already a huge breakthrough in the metastatic breast cancer field. So it, this is just a demonstration of how monoclonal antibodies and good research from Genentech has changed the treatment outcome of cancer patients. And this is standard therapy right now. Uh, uh, at this point, Herceptin Perjeta is still the first line for HER2 positive breast cancer patients. So as you can see, at 8 years, 37, which means 1/3 of the metastatic breast cancer patients are still alive. Think about that. You have a you have a stage 4 breast cancer metastatic diagnosed, and you start the treatment with Herceptin and Perjeta. After 8 years, still alive. This is really, really groundbreaking. Okay, so now that we hear uh, how amazing Herceptin is, we have to go back to our current theme, which is biologics. So before talking about biologics, remember that a few minutes ago I just mentioned that each biologic, their manufacturing course has a lot of differences, and these differences actually led to batch to batch variation. So it's kind of weird to think about how each different batch would have a different uh, molecule coming out. So, and this is even more difficult to imagine if you're in the the pharmaceutical industry. It's kind of considering that, you know, a manufacturing assembly line, you're expecting every drug that comes out of the manufacturing assembly line to be identical. That's what you would expect, right? Okay. But now here I am telling you that biologics, the manufacturing for biologics would have an interesting result or phenomenon that each batch, when we talk about batch, usually we talk about the terms of liters. So the whole factory will have the capacity to produce usually it's a couple of thousand liters, let's say 5,000 liters of uh, cell culture. Well, think of it. You're doing when you're doing in the lab, the scale is about 1 milliliter, 2 milliliters. But in the manufacturing industry, uh, the size is 1,000 liter, 2,000 liter. Uh, and then they extract the cells and they collect the protein, the biologic, or in this case, the antibody that's being made, and then they purify it after a very rigorous process of purification, and then that is the drug that is being injected into patients. So the consideration of each batch being different to each batch, we call it the batch to batch variation, is because if you're doing cell culture, you would understand every week your cell culture is a little bit different. This week, maybe it's a little bit, the media looks a little bit more yellowish. Maybe this week, the cells are a little bit too confluent. So that's the same with a large scale. You just think of the problem as being amplified. And that would lead to the differences in different batches of the monoclonal antibody that's being produced by these cells, and that is and Herceptin is an example. This is a table that looked at the ADCC. ADCC is basically a objective measurement of how good Herceptin can kill the cells. So don't we don't need to go into details. Just look at the numbers. In the batch, all the Herceptin that was produced prior to August of 2018, the ADCC is around 80, 90, 100. Afterwards, after 2018, the ADCC is about 50. And then afterwards, the ADCC is about 80. What does that mean? That means that if you have a Herceptin from a different batch, the results are actually different. This is kind of scary when you think about it, but it also reflects the phenomenon for the biologic industry. And it is the biologic industry is more like kind of like a whiskey company. So we know that whiskey or wine are from different years and would have different results and character would have different like flavors and testing and ultimately different prices. So like a fine red wine, the year of the manufacturing and where it comes from. It comes from France. It comes from Napa Valley, uh, or whiskey if it comes from different company, even from the same company. When it comes from a different year, the taste is different, and so is the price. That's kind of the way to think about biologics. So to characterize biologics, you need a lot of preclinical studies and as well as biochemistry studies. So as you can see, mass spec, antigen binding activity. There's some spike in results. There's some trypsin and proteomic studies. These are all important in characterization of the biologics. Another example also was reported for Herceptin that there would be increased in asparagine deamination. So basically, there's a modification of the amino acid on the Herceptin monoclonal antibody, and this is also kind of interesting and scary and interesting at the same time. That the different variants would actually lead to a different composition of herceptin in the bloodstream. Also, this is an interesting phenomenon. So, in the first 40, 50 minutes, we've discussed extensively about biologics, and I've shown to you the important clinical rules using important clinical roles using Herceptin as an example. So therapeutic antibodies are now important, very, very important in cancer. Biologics are labor and manufacturing extensive, high margin profit, and they are novel. And Herceptin, or this name is the academic name is trastuzumab, are prone to modifications, especially in vivo. And this is actually kind of important if you want to hope for the best efficacy clinically. So after we've discussed extensively about biologics, now we need to talk about biosimilars finally. What are biosimilars? So biosimilars are products that are highly similar to the reference product. Again, we just mentioned the reference product is a FDA approved biologic. Okay, so every biosimilar has a reference product. That reference product is a biologic and is approved by FDA, and it cannot have any major differences in the clinically active or inactive components. No clinically meaningful differences between the biologic product and the reference product in terms of safety, purity, and potency of the product. The bulk of biosimilarity is the evidence of proving that your biosimilar works, which in this slide they call it the bulk of the biosimilarity, which basically means that how do you prove that your biosimilar is similar, right? It has to be similar that you call it biosimilar, right? So most of the studies are biologically, I'm sorry, are preclinically. This is different to a new drug development, a biologic. A biologic, if you want to get FDA approved, the most of the work will be in clinical development. There's a lot of discussion on clinical development, and I have a whole course this semester, but this semester is about going to end anyway. In our institution, I'm from the Institute of Clinical Medicine. So my course is exclusively talking about cancer drug and their related clinical trials. So basically, the clinical trial development is in itself another whole huge issue and art. But if you're a company developing a biosimilar, then the you will spend most of your effort not on the clinical side but on the preclinical side, because you have to do a lot of preclinical studies, including mass spec, including other biochemistry measurements and assessment, and a lot of experiments as well, to make sure that your biosimilar is similar to the biologic, to the reference biologic. If you cannot prove this, you can if you after your characterization you find that your biosimilar is, let's say, it's better, it's not similar, it's better to the reference drug, then it cannot be called a biosimilar. It can only be called a biobetter. And in that case, you would go through the full development and the approval process. So that's very interesting. When you are taking a test, you want for the best score possible. However, in the world of biosimilar, no, that's not the case. You want it to be not better and not worse as well. You want it to be similar. That's the essence of biosimilar development. So, I think previously we had a slide about biosimilars and small molecules. So the difference between biosimilars and small molecules, I'm sorry, biologics and small molecules, are the differences between biosimilars and generics. So both generic and biosimilars have a reference, right? Generics are basically just exact copy of the small molecule, and biosimilar is the identical product to the biologic. However, there's a fundamental difference. Generics are usually made through a chemical process, It's basically a manufacturing line, which you would expect that every lot or every drug has to be identical. However, biosimilar is made from different biology processes. So as we previously just mentioned as well, biosimilars would be um, um, they have batch to batch variation. They have a different lot uh, consideration, and they would never be totally identical to biologics. So you would give the biosimilars, a range that is acceptable. This range would include the glycosylation pattern, the post-translational manufacturing pattern that you would think to be acceptable. That's how you get the biosimilar approval. So this is basically a table that's comparing biologics to generic drugs. And most of the differences are basically the difference between biologics and small molecules, right? So now you can understand why this class is talking about biosimilars, but I spend most of my time explaining biologics because that's fundamental to understanding biosimilars, right? So this class is about biosimilars, but you understand biologics, you understand biosimilars. So differences between biosimilar and generic drugs, you can see it's just the difference between biologics and small molecules. So the originator is another name for the reference biologic. The originator themselves would have lot-to-lot variation. We just saw the case of Herceptin. Herceptin is the trade name, so that is the originator. So if you make so Herceptin itself has lot-to-lot variation, and if you produce a biosimilar, you will also have lot-to-lot variation. Then it has to be well regulated. The FDA will not expect you to be 100% identical to in terms of the variation to Herceptin, but the range has to be acceptable by FDA. And if there's a more than subtle change difference in comparison to the reference, sometimes the FDA will even require a clinical trial to demonstrate that the difference will not change the clinical efficacy. Again, I want to stress the fact that biosimilars cannot be clinically better, cannot. They can only be clinically equivalent. We'll have an example in a few minutes. In 2009, there was a BPCIA law that was passed in America, and it was developed as part of the Affordable Care Act. And this provided the FDA to set up the pathway for biologic products. And this is now the main regulation for biologic uh, regulation in FDA. And each country or each organization, such as European countries that is regulated by EMA, China is regulated by CFDA. They will all have their own regulations regarding biosimilars. So biosimilars and generic drugs have different FDA approval processes. The 505J is used to approve generic drugs. Uh, and generic drugs, if you can make sure that the manufacturing process is exactly the same, is actually exactly okay, then you don't really need to show safety and efficacy. You don't need to set up a very extensive clinical trial. Uh, if you can if you can prove that it is identical in your manufacturing process. Um, However, the BPCIA would yeah, require um, a more detailed proof that the biosimilar itself is working. and is similar and is clinically equivalent to the reference product. So we just mentioned the FDA regulation. The FDA regulation looks at the totality of evidence. It basically, this is just a fancy term that says that FDA regulation for biosimilars will require a lot of studies, basically analytical studies, animal studies, and clinical studies. The analytical study is used to show biosimilarity, to show that the biologic drug is highly similar to the reference drug, and also you need to do the standard toxicity animal studies, and also and a pretty basic clinical trial. So we've mentioned this previously. Biosimilars use the same mechanism of action. It cannot have any new unknown mechanism, and the administration route has to be same. If your reference biologic is IV, then the biosimilar has to be IV. If you're developing something that's a biosimilar but has a different route administration, then more will be required of you for the at the clinical trial stage. This is a slide. There are some Chinese in here, but basically, this is all the different market, all the different market status of the biosimilars. So this is EMA, European Medical Association, and the there, as you can see, there's a lot of biologics that are currently being approved and being sold at in EMA. This is Korea, this is Japan, this is Taiwan, this is US, this is Canada. So Europe is basically a leader in biosimilar drugs. So these are all the academic names, and there are several biosimilars that are being approved in each of these countries or regions. So again, we just mentioned that biosimilars would require a very extensive clinical evaluation to establish that they're very highly similar to their reference, and all of the physical, the biochemistry, or the biophysical properties has to be identical or nearly identical, and then you need to show some functional assays. Again, sorry, this slide is in Chinese. Basically, the left is a cartoon figure of the development process of a new drug. The right is a biosimilar. And you can see the new drug, every step is roughly equally important. So you start with the CMC and the drug toxicity profile, and then the pharmacokinetics, pharmacodynamics, or the PK and PD. And lastly, you go to the clinical study. These are all important for new drug development. But as you can see for biosimilar, this is like a pyramid. The base is more wide, meaning that you would need more data from the CMC data, because you would have to provide a lot of data to prove that your drug has the same CMC and same physical chemistry, biochemistry characteristics compared to your reference drug, while as the requirement for clinical studies is much smaller. So a very short note on the how to call the biosimilars or the nomenclature. Basically, the name that comes first is the academic name of the reference drug. So adalimumab, which is Humira, is the name. Adalimumab is the academic name for Humira. Humira is the trade name. So any biologics that is based on adalimumab, the name would be the academic name would be the original name with a hyphen and 4 letters, and these letters can be random. Uh, so they can you can still call it a trade name, and that is what we everybody does. But the academic name will have to be similar. Well, not similar. It has to be identical to the academic name of the originator, and also added by followed by four letters. So that is interesting and important to understand the biosimilar naming. So this is the naming rules of the US FDA. This is the naming rules of the Japan FDA. So basically, a little bit more, a little bit longer, the academic name, and then BS stands for biosimilar, and then the infusion dose, and then the name of the company. So we're going to talk a little bit about extrapolation. This is an uncommon, not as common, but it's something to be considered in biosimilars. So let's talk about the general concept. Every drug company, their first priority is to, well, make money, right? That's every company's mission. You have to survive before you can save the world. So if they spend all these years developing a drug, they wish the drug can get approved in as many indications as possible. But in general circumstances, each approval has to be in a clinical trial for each separate indication. So if this drug has done a clinical trial for breast cancer, then it gets approval in breast cancer. But you cannot use it in gastric cancer. You have to do a separate trial in gastric cancer. Now, this is important. It's a the following explanation is important to understand extrapolation. Okay, uh, let's say a originator, a reference drug, has three indications in three different, or one indication but in three different cancers. That's basically three indications. So it can treat metastatic breast cancer, gastric cancer, and colon cancer. Let's assume this is this drug. If you have a biosimilar and you prove that it is working, it is efficacious in just one of the indication, you could apply to, you can bargain with the FDA and you say that, okay, I've demonstrated that my drug is biochemically and physically similar to the reference drug, and I've demonstrated that it is not better or not worse in breast cancer. So I would like you to give me the indication for not just one cancer, but all three cancers that are approved for the reference. I hope you understand what I'm saying. So for the reference, there's no question. Each indication needs a separate clinical trial. But for biosimilars, you just need to do one trial, and then you can get an indication for three. But you cannot get an indication for uh, an indication that is not approved for the reference. That doesn't make sense, right? But for those that already have indication and you prove that you've already proven that your drug is similar to the reference, then yes, you can negotiate. There's no guarantee, but you can negotiate with FDA and they would give you the indication for all the other cancers. That's called the extrapolation. Okay, this is just a summary of what I've just mentioned and what's the consideration for FDA. So this is another kind of similar pyramid illustration. So as you can see, for biosimilars, the weight is in analytical and preclinical, and for the reference drug, the weight of development is in clinical studies, and this is correlated to the cost that is spent. Europe is the global pioneer in biosimilar acceptance. It has been currently approved the most biosimilars, and they adapt biosimilars pretty rapidly. So as you can see, uh, trastuzumab has was approved in these indications in different timelines. So there's now several trastuzumab, which is Herceptin. You remember Herceptin that we were just talking about like 20 minutes ago? There's several biosimilars that are being approved in Europe, and also another biologic drug called bevacizumab. There's also several biosimilars. And the breast cancer guideline uh, produced by ESMO, which is the European Society of Medical Oncology, supports the use of biosimilars. So this is really important that basically the European Medical Agency endorses the widespread use of biosimilars. Now, for the last few minutes, we will discuss a trial, a clinical trial. This is basically you can see how a biosimilar is developed, and we use the biosimilar to uh, HER2 antibody reference drug, which is Herceptin. So we use the Herceptin biosimilar, and the name is called Ogivri in HER2 breast cancer. This is the preclinical evidence, and as you can see, this mass spec data, the above is of Givri or the biosimilar, and the below here is Herceptin. You can see all the peaks line up and are very identical. It, of course, it will not be 100% equal, but it looks very identical, and you cannot have any new peaks uh, in this figure. This is the structure that is proven that the binding is the same, and this is also DSC to prove that the binding pattern is the same. And here we can see all these checks. They do a lot of preclinical analysis to make sure that the biosimilar is similar to the reference, which is Herceptin. Then they do a phase 1 trial. So you can see they use the European Herceptin, they use the US Herceptin, as well as their biosimilar. So they do a lot of blood samples examination at different time points, and the all three P PK curves are lined up perfectly. So you can see there's basically no difference in these curves. Then they do the phase 3 trial, which is a head-to-head comparison between their biosimilar and Herceptin, and they will have to prove that clinically the efficacy is equivalent. So this is the baseline characteristics for all the patients who are enrolled in this phase 3 trial. And the primary endpoint is the response rate, which is a measurement of how much the tumor shrinks. And you can see that the drug Ogivri response rate is about 69%. Herceptin is about 64%. So then you can argue that, well, 5%, is that a significant difference? They will run statistics and they will show that, okay, it's not, the p-value is larger than 0.05, so it's not significant. So then statistically you cannot say that there's a difference between these two groups. And the ORR ratio, which we're not going to detail how it's calculated, this is for the clinical trials class, but basically the ORR is 1.09. The confidence interval overlaps 1, so you can say that there's no difference between the two groups. And then the survival endpoints, PFS and OS, are the curves, as you can see, are very, very overlapping. So this is very identical. The p-value for both cases are not significant. So the biosimilar trials are those that you would wish to see a non-significant p-value. Usually in biomedical research, you want to see a significant p-value, but this is one of those cases that you really don't want to see a significant p-value. So again, progression-free survival at 36 months, no difference. So you would say, isn't that to be expected? Well, this is how clinical trial works, because anything that is approved to be given to human, you need to prove that First, it is safe. Secondly, it is efficacious. If something is not, you cannot prove that it is working, it is efficacious in treatment, then even though it is safe, it should not be approved. So, clinical medicine is one of those scenarios where uh, you cannot only prove that it is safe. You have to also prove efficacy for a drug to be approved. So that's why new drugs to be approved is actually very difficult. Uh, I don't know if you know the number of new drugs that are approved by FDA each year. It's in the range of like 20, 30, 40. And you would think that for all the indication, all the diseases, uh, including like oncology, infectious diseases, you would think that you, every year there would be more than only like 30 new drugs per year. But no, only a couple of dozen new drugs are approved by FDA, and thousands of drugs are being actively developed. Only just a handful or two or three dozen can be approved. So you can see how difficult the process is. So overall survival, no significant difference. Significant difference. And the safety profile, as you can see, each row is represents the incidence of a safety issue: alopecia, neutropenia, neuropathy, etc. And if you compare between the two arms, the numbers are not going to be exactly the same, but you can say that it's not that different. And in terms of the incidence of the adverse effects, it's comparable. LVEF, left ventricular ejection fraction, is a measurement of a cardiac output or cardiac function, and is identical between the two drugs. Immunogenicity, this is just a measurement of whether the drug will cause immunogenicity. So as you can see, uh, both trastuzumab and the biosimilar will cause some ADA uh, uh, after treatment. So this is kind of interesting. However, the incidence is really low, and the biosimilar itself does not add any new incidence of causing anti-drug antibodies. So it's similar between the two products and safe, that low immunogenic potential. So in summary, the biosimilar Ogivri has shown preclinically and also clinical data that is comparable to U.S. and European trastuzumab, and therefore it was approved. So these are now the current major Herceptin uh, biosimilars. As you can see, different produced by different companies. Mylan, Pfizer, Amgen, and Samsung and Celltrion are actually both South Korea companies. So kind of interesting that South Korea is now an important leader in biologics. So this is an interesting breakdown that you can see. This is the European market, different countries, and you can see like this is the market share of the Herceptin biosimilars. Once the EMA or European Medical Agency has approved Herceptin, uh, almost 90% of Denmark shift to using the Herceptin biosimilar. Different companies, different countries would have different rates. Like this country, it's probably Slovenia. Slovenia has a very low rate of Herceptin biosimilar use, but maybe it's policy related. I don't know. But basically, you can see that European countries are really friendly and willing to use biosimilars. So um, to summarize, biosimilar is a really important trend for the biologic new drug development because biologics are really expensive, and but then a lot of the major biologics like Herceptin, we just saw the history of development of Herceptin, and the patent for Herceptin has now expired. So now Herceptin biosimilars are available on the market, and they provide the patients with a more accessible, more cheap, affordable choice while being uh, assured by the FDA that the preclinical and clinical efficacy is comparable. So I think that's really important. So clinically, as a clinician, for us, it's really I think it's helpful to the patients. They're definitely benefiting from the introduction of biosimilars. So this is definitely an important trend for the pharmaceutical industry development. So thank you all for your attention today. And again, my apologies for not being able to deliver this talk in person. Please do not hesitate at all to contact me if you have any questions about this class, about biosimilars, biologic development, or in general, if you are interested in cancer drug development, cancer research, please feel free to contact me. My lab is working basically on breast cancer development, and we're working on a molecular biology mechanism, but we are always geared towards therapeutic development. So it's actually very interesting that we recently discovered a mechanism, and just recently started to partner with some uh, pharmaceutical companies. One is in US, one is in Korea, and we are starting to test their compounds for this mechanism. So it's actually a very interesting approach, and I think that is the ultimate goal for biomedical research, that we want to have a translational impact for our basic researches. So that's it for today's talk. Please let me know if you have any questions. Thank you.